All right, I guess we can get started. I'll just very quickly introduce Nenad. Thanks everybody for coming back to the session. Again, it's my great pleasure to introduce Professor Nenad Pavin, who is a professor of physics at the University of Zagreb in Croatia. He's now going to expand on Eva's talk by going into the theory behind mechanobiology of the mitotic spindle. Nenad, go ahead, please.
Okay, thanks, Vaish, for this, for inviting me to this great meeting and organizing everything. It's a real pleasure to talk to all of you. So, as already Eva introduced, I will discuss mainly mechanics of mitotic spindle. And before going there, I will briefly introduce some physical basics of the cytoskeleton. Then I will go to mechanics of chromosome positioning in the spindle, how they are getting aligned to the metaphase plate. And finally, I will, the rest of the talk, I will discuss rotational forces and how these twisted shapes are generated and how we understand the shape of the spindle in general. So when we talk about the physical basis here of the cytoskeleton, and in principle physical basis of the of some cell biology in general, there are two reasons why I need to discuss this in more details. One is just to answer why we bother biologists with this physics, and why biologists will need to also to learn something about the physical basis of this cell mechanics. And the other reason is I need to introduce the physics that is actually that one can apply to the to these cells that are of micron scale, and obviously the physics that we learn and that we actually somehow experience in our macroscopic lives maybe will not be easily applied to the physics at the cellular level. So when we check cells, we can see something really striking. They are neatly organized. They have organized interior. For example, here you can see this beautiful spindle that is positioned within the cell with respect to the cell cortex. And you can see nicely organized microtubulars. Obviously, a lot of self-organization is going on there. Also, one can see these part proteins that are bound either to anterior or posterior side of these cells. And when we see this self-organization that happens at the cellular level, one can ask what are actually principles or what strategies are behind this self-organization. Self-organization relies on several strategies. One is transport of material. If you transporting some material, you can use diffusion as a way of transporting material. For instance, if you have more molecules at one side, by diffusing, they will eventually getting uniformly distributed. But also, you can also see that sometimes cells are using filaments to transport some cargoes along these filaments by molecular motors. Besides transporting material, cells need to measure distances, and also here there are at least two big ways how cells can do it. One is measuring gradient of molecules. For instance, I will now point the same this scheme related to the diffusion as before. So if you have source of molecules on one end and sink on the other, there will be a gradient of these molecules. And if one measures the concentration, local concentration at a certain point, he or she will actually measure the position within the cell. The other way to have or to measure distances is to have gradient of some molecules along filaments. So if you want to measure just what happens along the filaments, not in the entire volume, so you want to see, for instance, this locally, how this microtubule is long, you can apply something what's called so-called antenna model. So these molecules are binding with equal probability along the microtubule lattice, but by moving processively towards one end, there will be more or larger concentration of these motors, the longer microtubule is. So these are two ways how distances are measured here in cells. And finally, I will not show the drawing now, but the rest of my talk is mainly related to the third part of or physical, some physical scenario related to self-organization is generating forces. So generating forces relies on the cytoskeleton, which is some filaments like microtubules and motor proteins. But before going to the cytoskeleton, here I would like to show you one example that is very simple, and you only need high school physics to understand what I'm discussing here. So just imagine that you are swimming in the sea and you stop waving your hands. You will continue to move through water, but also you will stop because of the friction with the water. You will stop at a certain distance, and you will cover a few meters before you completely stop moving through the water. And here one can ask, okay, bacteria is also swimming through water, and what distance this bacteria will cover before it stops. So bacteria, for propelling through the water, actually uses this flagella, and by beating this flagella, it is propelled through the water. However, one wants to calculate what the distance it will cover when it stops beating, and here is the very simple equation. So it is the mass term times acceleration plus the friction force, and this is Newton equation for the system, very simple. And if you solve these equations, you will get this exponential behavior of velocity. So velocity decreases exponentially, and you can also calculate typical time for stopping here. So the typical time is the ratio between mass term and this friction coefficient. Interesting part appears when you put some numbers here. If you put some numbers and you calculate this tau, you will see that actually this tau is 0.2 microseconds. So this bacteria will stop moving on a microsecond level. And if you multiply this by velocity that initially it had, you will get the distance that bacteria will cover. And we are now getting the length of 0.05 angstroms, what is actually the distance of 5% of atom size. And so if we check this distance covered by this atom size or this bacteria, we can see that it is indeed very small distance covered, which brings us to several interesting conclusions. One is actually if one wants to calculate how calculate the movement of these bacteria, in principle, one can just ignore inertia in his or her equations. So what is typical equation for describing processes on the cellular level are force balance equations. So we have here friction force, and we have some external force that actually exerted to this bacteria, and these two forces are in balance. And of course, don't be confused that I'm now saying that Newton equation is wrong. No, this is completely, this comes from Newton equation, so everything is fine. But only the equation of motion now is not having the inertia term, but has only these two terms, and this help us in calculating some not so simple cases as was this one. And even more important thing I want to point here is already this very simple equation, one can see that our senses are not always good enough. So what we built in our macroscopic world, maybe it's not can easily be applied on the cellular level. So one can say our sense can deceive us if we really want to apply just macroscopic ideas here in the microscopic world. So to generate forces, to organize its interior, cells have cytoskeleton. So what is the cytoskeleton? We've already have shown this movie where you can see filaments. These filaments can grow or shrink. Also, these filaments are now here used as a track for this motor to move along this filament and to transport a cargo along it. But this was only on some phenomenological level. Here I would like to introduce ideas how the forces in cells are generated and how these forces arise from some basic physical principles. So first, I would like to explain how motors are working. For example of principle of motor working is here non-processive muscle myosin. This muscle myosin, here you can see how it's going. Muscle myosin is the non-processive motor which binds to the filament, exerts some power stroke, and then unbinds from these filaments. And because it in principle performs only one step, it is so-called non-processive. This non-processive motor is well studied. So what we know that in principle it's working in 4 basic steps. First step is here with the motor head bound to the filament and ATP to it, and it is at certain conformational shape. When it releases this ATP, there is working or power stroke, so there is conformational change in this motor, and by this conformational change, this motor performs work. Then to unbind, the motor binds ATP, so now the motor detaches from the filament, and then finally there is a recovery when there is hydrolysis. So instead of ATP, now here in this motor head there is ATP plus phosphate, and finally when this motor head binds again to the filament, this cycle completes. This is how non-processive motor is working. Very similar is physics of processive kinesin. You can read about this. I will not go into details. or you can watch this movie while I'm talking. So what is the principle? What is the basic difference between processive and non-processive motors? Is that these processive kinesin motors are performing many steps along this filament before finally detaching from this filament. Of course, now we saw that motors are moving along the filament, but it's question what happens with these motors if some cargo binds to it. Meaning, motor, as I initially mentioned, transporting cargos along the filament, and now we have motors bound to some cargo moving along the filament. If there is no any force applied to this motor as opposing force, so meaning there is no cargo, this motor will move with certain velocity. Here on this graph, the point is here. However, if you now apply some opposing force, because of this opposing force, motor will have slower and slower velocity. This behavior, or this characteristic of motor is so-called force velocity, and these force velocities are studied for many motor proteins, also including kinesin 1 shown here. Also, the question is, yes, we have this phenomenologically shown force velocity, but how we understand this movement of motors along the filament from the physical principle? One can draw a free energy landscape for kinesins like this one, saying, yes, we have kinesin bound to a certain point at this lattice, and then this kinesin can move in one or in the other direction. For moving to the right, or when it's moving to the right in this scheme, it consumes energy, it consumes ATP energy, and thus it's going to the lower local minimum, whereas it is going to the left, it actually needs energy, and this is now going against this energetic landscape, and obviously that chemical rate for moving to the right is much higher than the rate for moving to the left. Also, if you know these chemical rates, one can also calculate velocity by its moving from along this filament by hopping from one side to the other. You can see that these rates are just here given by Boltzmann distribution. So what happens with these rates if we now apply some forces? This is just a landscape with two minima. The first minima is here at x1, the second one is x2, and of course there is a certain rate of going back and forth here along this landscape. But what happens with this landscape if we apply some force? So if we apply some force, then this landscape changes. So a slightly bigger force, we are getting this blue curve, and even bigger one, then we are getting this green curve. And obviously, because this change, the change in this landscape, it will also influence the chemical rates of hopping from one minimum to the other. And if we now make a link between these rates, forces, we will get actually the force velocity relationship for one state case of motor stepping. Here I need to say that this relationship is has some general shape, and it is rather complicated and non-linear. However, if we want to calculate something, we usually use a linear approximation for this force velocity, as it's written here. Why linear relationship? It makes life of our theoreticians really much more easier, and I think many calculations won't be possible if without this approximation. Of course, so much about motors, but also there is a question: what happens with microtubules, and can microtubules also exert forces? So microtubules are dynamic filaments that can grow and shrink, and they also have some tube structure. This will also be important for something what will appear just after I explain how microtubules generate forces. So what happens when microtubule is growing? As long as it's growing freely, it is growing with certain velocity, but also when it's growing against some obstacle, it keeps growing by exerting a pushing force against this obstacle. And can we understand this can we understand these pushing forces of the growing microtubule? For understanding this, we also need to just recall that we are in the microscopic world, and that sizes are different. So what happens there, and how microtubule can grow against barrier, even though it pushes against it? So in our macroscopic life, we would say, yes, there is a force pushing against some barrier and nothing can get in between. However, here, if we have this pushing force, we also have thermal fluctuations. We have thermal forces, and because of these thermal forces can make a gap between the barrier and the microtubule end. And how big energy is necessary for opening this gap? But that's just the energy that is equal force times this size of the gap, and because all these processes happens due to thermal forces, one can calculate from Boltzmann distribution probability for having this gap open. Of course, this is something very non-intuitive because we don't see this on macroscopic level, but as I mentioned at the beginning of my talk, our senses can deceive us, and that's why I'm pointing this once again. So here I would like to emphasize everything what we know from macroscopic world. We need to think twice before implementing to cellular level. Of course, apart from pushing forces due to growing microtubules, when are shrinking, they can also exert pulling forces. But I think he already mentioned this, and I will not go into many details here. So I have shown here these pushing forces. I have shown this microtubule being curved, but what one can see from this type of experiments, so the microtubule was growing against barrier, it's not only that it exerted a pushing force, but also one could see that microtubule is getting curved, and this curved shape happens because of this force, and it is so-called buckling force. To understand how the forces can change the shape of elastic object, I'm here first showing something what one can do with a sponge. So one can play with the sponge, and depending on how it applies forces, how someone applies forces to ends of this sponge, it will change its shape. So one can say, yes, we can, if we have this type of bending moments, we will have C shape. If we have the different bending moments, then shape can get also S shape, or eventually one can get twisted shape. And what one can see also from this example, it's quite obvious that if we know the shape, actually we know what happens on these sides, and vice versa. If we know what type of forces we are applying here, one can also predict shape of this object. In the most simple case, if we are now checking what happens with shape, that with an elastic object that has a bending moment applied to its end, it will get a circular shape. So this will be a circular arc with a curvature 1 over radius, and this curvature is actually related to the bending moment and its rigidity. So here one can see these three different physical quantities that are mutually related, and if we know two of these two quantities, one can actually determine the third one. Here is one nice example what was done for measuring rigidity of polymers. So here is a microtubule that is perturbed by thermal fluctuations. We know how thermal fluctuations big are, so one can actually know these bending moments I mentioned before, and same for the actin filaments. So we can also observe their shape, and because of their shape and known bending moments, one can actually calculate bending rigidity of microtubules and actin filaments. Of course, this was so far about what happens on a single protein level or on a single filament level, but in cells, actually also bigger forces are generated, even macroscopic forces that are also driving our muscles. So if we have different tasks to perform, actually cells uses different strategies. First, if it transports objects along some small objects, it can transport them along the filament, like walking along the street. So there is one motor moving along this track, and by moving along this track, it actually transports object from one end to the other. If objects are larger, then it's not one motor enough for transporting them. Then multiple motors are bound to microtubule, and then these multiple motors can exert collective forces, like these guys pulling on this rope, and then because there is more of them, they can move larger objects. And finally, there is also a situation when more filaments are cross-linked, and by cross-linking filaments, I'm sorry, by cross-linking filaments, these motor proteins can also exert forces. So what happens if we want to transport small cargo? In this case, we have a single motor situation and in the single motor situation, we can describe it by force velocity relationship, which I already mentioned. Here I'm again repeating this linear force velocity relationship, which I will use later in my talk. Of course, there is a case with more motors that are transporting objects. If there is more motors, then these motors are now exerting collective forces. These collective forces depend on number of motors bound to the filament, and also they depend on the force exerted by a single motor. So here, to conclude my talk, I would like just to repeat what I mentioned before. So if we want to understand the elastic objects that occur due to some bending moments, we can do just by applying the elastic theories. Then we also saw that motor proteins are exerting forces that are of size of several piconewtons. And finally, these motors are organized in a different way to perform different tasks, thus depending on the cellular process that needs to be fulfilled. So here I'm happy to answer your questions.
Thanks, Nenad. Are there any questions for Nenad at this time? If not, perhaps I can ask you a question regarding active transport in living cells. Given the crowded environment in a living cell, how likely is it that even small vesicles can diffuse to get around to locations inside the cell?
Can you repeat the question? I'm not sure that I completely understood your question. So I mean, you have a small cargo that is diffusing around.
Yes, and then I'm wondering even something of the size of, let's say, 100 to 200 nanometers. That's relatively small when you compare intracellular cargo, such as organelles. I'm wondering if there's evidence to show that even for such small cargo, diffusion is constrained because of crowding, because of, let's say, the actin meshwork, or even the microtubules that are present inside the cell. And so how would this then, if it's possible at all for diffusion to be a mode by which even small cargo can actually move around in living cells?
Depends on which cargo we are talking here. So if we consider just some proteins, not even their complexes, yes, they are diffusing around, and because of diffusion, they can bind to bind to its partner. Also, some aggregates are diffusing around the cell, and as you mentioned, smaller this cargo is, its diffusion is faster. I'm not sure when these objects are too large to get actually stalled by a mesh of actin filaments. That I cannot answer.
All right. Thanks, Nenad. We don't have any other questions at the moment, so maybe you can continue with your talk, and I urge the participants to put in any questions that they have in the chat box, or raise your hand if you don't feel like typing. Thanks.
Okay, so what I have shown before is some general principles how microtubules and motor proteins are working together to position some organelles. Here, I will discuss now what happens during the cell division when chromosomes are positioned to the metaphase plate, and what forces are working there to transport such objects and get them properly positioned to the center. What happens during metaphase in spindles. So here is one metaphase spindle, and one can see nicely organize these kinetochores and microtubules with chromosomes being almost ideally here placed to the metaphase plate. And of course, there is the question why we want to study such process. So it is known that if we have them properly aligned, that everything is working perfectly. However, if these chromosomes are not really nicely aligned to the metaphase plate, then there is a failure in a cell division, and thus it is really important for cells to get chromosomes properly aligned or congress to the metaphase plate. What we know so far about kinetochore positioning or chromosome positioning, we know that there are forces important for this process are generated first here at the link between microtubule and chromosomes. This is this magenta kinetochore here, and these forces are pulling forces by this microtubule from one side and also from the other. Then there is microtubules that are not bound to the kinetochore, but they are now pointing to the chromosomes, and because they are now linked to the chromosomes, there is a pushing force generated by motor proteins here at the chromosome arm, and this is so-called polar ejection force. Here I would like to emphasize that this force is so far considered as the main centering force in mammalian spindles. And finally, also apart from having these microtubules being pulling from one and the other end, one actually needs to regulate which microtubule will pull stronger, or from which side it will pull stronger, to have actually centering forces by pulling forces in that case. So what happens in this third case? This third case are length-dependent accumulation of kinesin proteins that are accumulating at the plus end of the microtubule, and this kinesin, this third scenario is working together with the first scenario, meaning these proteins are regulating the microtubule dynamics, and more protein accumulated here will generate faster depolymerization of this microtubule, meaning it will generate more pulling forces from that side. However, this picture, as Eva already mentioned, is not complete. So if we check what we do have here, so we have two kinetochore microtubules bound to kinetochores. It's not visible here, but I think it's quite clear when the kinetochores are. And then in addition to these kinetochore microtubules, there are bridging microtubules that are extending and connecting these two kinetochore fibers from opposite sides. Here, they're drawn in a more schematic way. And I would like now to discuss how the bridging microtubules actually affect the centering of kinetochores. If we have this picture in mind, we know that these microtubules that are bridging to kinetochore fibers, they will somehow influence the forces acting on the kinetochores. But to understand the mechanism, I will introduce a model that is more simple than this scheme shown here. So what I'm showing here is the one-dimensional model with kinetochore microtubules, these upper microtubules, and bridging microtubules, these lower microtubules here. And now I will show you the main ingredients of the model to get idea how the motor can work. What forces are present there? First, what we have is this link between the microtubule and kinetochore, and this connection, the forces here exerted by the microtubule and the kinetochore are described by the force-velocity relationship. Elastic property of this chromatin that links to chromosomes, that links to chromatids is described by hook and elastic spring. Then we have motor proteins that are pulling at the pole, again described by force velocity. We have also these motors that are regulating depolymerization, and finally we have cross-linkers, motor proteins that are cross-linking antiparallel microtubules, and passive proteins that are cross-linking parallel microtubules now. What I emphasized here is just, I wanted to emphasize that cross-linking proteins, there is a collective behavior of them, which is described here by this simple relationship, and also there is some force exerted by these passive elements, which is again described by this equation that tells us how the force of multiple passive cross-linkers are. Of course, in addition to these ingredients, one needs to write down force balance equations for each element here. What I'm not showing because it's rather technical, and I think it's not easy to grasp basic ideas from that. So what we do have here when we introduce such a model, first what appears is a length-dependent force. So there are motor proteins here that are cross-linking antiparallel microtubules. So here is one antiparallel microtubule, here is the other antiparallel microtubule, and there is more motors here on this side. In this initial case, antiparallel region on this side is even not formed. Because of this length-dependent force, the net force on these chromosomes and kinetochores is towards the left, towards the right. And now, how we can visualize what happens in simulations. So what we have pulling forces from right side that were larger compared to the pulling forces from the left side, and because these forces win, we are getting movement to the right. And here I'm just showing now the size of these two forces, of these two opposing forces. To make it more clear, what we do have here, so we do have here these cross-linking proteins that are accumulated here in the length-dependent manner, meaning more larger this overlap is, more motors is accumulated, and bigger force is generated. And of course, now we have a situation with shorter overlaps. They accumulate less cross-linking motors, and then these forces are getting smaller. Of course, I'm showing here forces now from both sides. We see as kinetochores are approaching to the center, these forces are getting closer and closer, and when kinetochores are in the center, these two forces are equal. In addition to these active proteins or motor proteins, there are also these passive cross-linkers. These passive cross-linkers are now linking not anti-parallel regions, but they are linking parallel regions like this one here, or parallel regions like this one here. And again, because of this difference in their length, we will get different forces. We will get larger forces to the right compared to the left. And again, only these passive crosslinkers can also actually move these chromosomes toward the center, and thus they also can actually be a centering mechanism here. Okay, so far about ideas. Now I would like to show you something what happens when we check this movie, and what are actually the outcome of these calculations. If we check what happens here during this chromosome positioning, we can see that the bridging, the kinetochore fiber and bridging fiber, they are both undergoing polar flux. But also we can see clearly from this animation that polar flux here in this bridging fiber is much faster than the one here in the K fiber. Of course, this difference is always present, but when kinetochore fiber is larger, actually this polar flux is closer to the bridging fiber, flux of the bridging fiber, whereas this K fiber is getting shorter, then this K fiber slows down. So this is the first prediction of the model, and what is also more important to emphasize here, it's not only the prediction of this model, it's key for getting efficient centering. If there is no this difference in polar fluxes, actually there is no centering mechanism by these length-dependent pulling forces. And thus, this is the key prediction that should be tested by the model. I think you already heard by Ineva's talk that indeed we find in experiments faster movement or faster sliding of this bridging fibers compared to the kinetochore fibers. Of course, one can play with proteins, or one can play with a number of these cross-linkers here, and ask what happens if we increase number of these passive cross-linkers here. If we increase the number of these passive cross-linkers, already on intuitive level, is getting more clear that the connection between K fiber and the bridging fiber will be stronger, and because of this stronger connection, these two objects will not slide as so much as they slide now, and because of that reason, there will be smaller difference in velocity on the left and on the right, and also we'll get a less efficient centering. So this is something what is done in Eva's lab. So they have shown that overexpression of these passive cross-linkers that are linking parallel microtubules are actually leading to less efficient centering and faster movement of the K fibers. So to conclude this part of the talk, I just would repeat what I have shown before. So we have shown that kinetochores and bridging fiber are fluxing with different velocities, and because of this difference in their velocities, they actually resemble the centering mechanism. And also what I have shown, that more passive cross-linkers leads to less efficient centering, and something what is also quite clear from this mechanism. This mechanism relies on these bridging fibers. So if you somehow perturb bridging fibers, you will get less efficient centering or off-centering of kinetochores. So here is the end of my second part. So, and we can also, I can also answer your questions here, or if you want me, I can resume my talk.
There are a few questions, Nenad, so maybe we can go through them. The first is from Sandhya Kaushika. Sandhya, please go ahead and unmute yourself. You can ask the question.
It was very nice talk, Nenad. I just
thank you.
Really appreciate it. As an experimentalist, I appreciated how carefully you went through background. In your model, you show that your motors essentially step in sync with each other. But in vivo, that may or may not happen. So in your models, if they don't step in sync with each other, would the bound motor, which is not working, be ripped off by this tree?
So this is schematic presentation of what happens here. And thanks for this question. Maybe I should also emphasize this, that this is just to somehow show how these motors are stepping here. Whereas when they are performing, of course, they are not doing this this stepping as I have here shown. So they are not so well correlated, and some motors are stepping faster, the others are just stalled, so waiting for some time before step. So it's
but the microtubule with even some motors moving will begin to slide, right? So the ones that are bound, would it sort of rip them off the microtubule? I don't know. I'm asking because I really don't know. It's not my idea.
Who will rip off? Sorry.
Because it seems to be bound to both to two parallel filaments. Yes. And so if one end of it moves, then it potentially the filaments need not move in sync in a manner which is suitable for the motor which is not stepping at that time.
Yes. Okay. Now I understood your question. Okay. Yes. Okay. The motors are so they can step faster. They can step slower, and also it is studied already for a while how detachment of motors from the microtubules depends on the forces, and indeed there is a force-dependent detachment of motors, and in general, in many cases, motors that experiences larger forces detach faster from microtubules. But if this is working properly, if the mechanism is working properly, the motors will detach from microtubules, but the other will bind from the cytoplasm and replace those that are that left microtubules. So there is always this this interplay between attachment and detachment of motors. It is not static as in my movie.
Thank you.
Thanks, Sandhya. The next question is from Saptarshi Chatterjee. Please go ahead, Saptarshi.
So hi Nenad, great talk. Hi. So I was wondering that if you incorporate the a length-dependent catastrophe of the kinetochore microtubules, then what happens to the positioning of the chromosome?
Yeah, thanks for the question. I didn't show the results for length-dependent catastrophes because it's already this discussed in the field. Yes, when we use length-dependent catastrophes, we are getting better positioning. What we are now discussing, and we are really not sure whether we are talking about very dynamic microtubules in sense that undergoes this dynamic instability, or microtubules are always growing faster or slower. It's so our experiments are not so conclusive whether these kinesin-8 motors are actually regulating the growing velocity rather than catastrophe of microtubules. We really rarely see kinetochores that are actually moving along the shrinking microtubules. Even if the kinetochore is moving, for instance, towards the pole, quite often its velocity is similar to the polar flux, and in principle, the kinetochore is not moving too much with respect to the microtubule.
Okay, I understand. Another question I have, that is, in the simulations, the motor binding, unbinding, binding, are those things explicit, or they have chosen from a mean field expression or something? So is there explicit?
So I'm using very simple description of the number of motors, which says that number of motors is proportional to the overlap length. So this size of this overlap sets number of motors. So I say there is uniform linear distribution of motors, and I multiply this linear distribution by the length of the overlap. That's how I calculate the number of motors in the cross-linking region.
Okay, thanks, Nenad.
Thank you, Nenad. We'll maybe take a couple more questions. Next is from Debashish Chaudhary. Debashish, please go ahead.
Hi, Nenad. Nice talk.
Thanks.
So, I have questions about two things. One is about the passive crosslinkers in your model, and the second one is force generation due to the motor, how you incorporate it within your model. So first question is that by the passive crosslinkers, what is the direction of force generation by the passive crosslinkers that you have incorporated?
So direction that I
crosslinkers, you have this. So how do you model that force?
So how do I model? So technically, I model this as a passive friction. I say that, so I just need to go to this. So I say that this crosslinker will exert force that is proportional to the relative velocity of these two filaments. So bigger relative velocity is, the bigger friction force appears here. And this is something what is also measured by, I think, Joe Howard. They have shown that that actually
that exponential growth in friction. Okay. The other part of my question is that in your model for forces generated by the molecular motors, do you just invert the force velocity relation?
Do I just what?
How do you define the force due to molecular motors? Is it?
Oh yeah, it's just I mean, I just use this equation to calculate the force if I know velocity. So here is the velocity as function of force. I'm calculating now force as function of velocity. Yes, you are right.
Inverting that equation.
Yes.
Okay, thank you. Thank you.
Thanks. And maybe one last question before we let Nenad continue. So Amrita Vasudevan, please go ahead.
Yeah, hi Nenad. This is a great talk. I just had a
Thanks.
quick question about the mechanism of centering of the kinetochore. Are you completely ruling out the role of polymerization of microtubules in centering, or do you just expect them to be same at both ends?
I'm not sure that I understood your question. What do you mean by ruling out polymerization?
So, since these microtubules would also be polymerizing,
Yes.
would that not contribute to centering of the kinetochore?
Uh, so what I'm using here is a description of what happens at the microtubule plus end. At the microtubule plus end, I will just go back to make clear this slide. So what I'm saying here for this link between microtubule and kinetochore is that the velocity of polymerization depends on the force. So if you have force applied to this direction, you will make this polymerization faster. And because of such relationship between force and velocity, I think I calculate this. I properly describe what happens there.
I see.
Is this your question? I'm not sure.
Yeah, so I was talking about how the relative polymerization rates affects the centering. So yeah, this addresses. Thank you.
Yeah, yes, and I just maybe just one sentence about all this model here. All these velocities are relative, and everything moves somehow relative to each other, and it's total mess when we write down these equations. So it's we are always one always need a lot of caution with science here. So, okay.
Thanks, Nenad. I think we'll let you continue with the rest of your talk.
Okay, and finally, this is something about rotational forces and twisted spindle. Here is the drawing how we understand the shape of the spindle, where we say that spindles are not some the microtubules are not some planar objects, but they are also somehow twisted. So let me explain how we are getting this and how we understand what happens there. So as already mentioned, we have these microtubules that are exerting forces on kinetochores. We have these overlap microtubules. So we have these K fibers, we have these overlap fibers. And I also have shown, and I will now just recall what I was saying before related to the shapes of some elliptic object that based on how these objects deform, we can actually infer something about the forces in these objects. So what actually what we would like to understand what forces act in these K fibers or in the spindle in general. And Eva already mentioned that there is this paradox that was already for a while in the field, that here close to the kinetochores, these kinetochore fibers are under tension, whereas here these microtubules are under compression. What brings us to paradox, because some object cannot be at the same time under compression and under tension, and to have some model that can make a mechanical explanation of how these microtubules are positioned in the spindle and how they are getting this curved shape. We had to introduce bridging microtubules, and now I will discuss how actually we found these bridging microtubules and what we learn from theoretical studying elastic forces here. The theory of this object is now more simple from motor perspective. I will ignore completely motor proteins. I will completely ignore force generators. And I will only now consider these microtubules as elastic objects that bend under some bending moments. So if we have some rotational forces here in this acting on some elastic object, these rotational forces will bend that object, and that object will attain some curved shape. The relation is simple. The relationship describes curvature and bending moments and this bending rigidity, as I already mentioned. And what we also can say that this shape can be generated by in two different ways. One is one can assume forces that are actually pointing towards the center, and these buckling forces can generate this shape, but also this shape can be generated by bending moment acting on both sides of these fibers. Of course, these two forces, or these two different boundary conditions will somehow produce different shapes, but these shapes are the difference between these two shapes are so small that cannot be experimentally verified. So in principle, we cannot say whether we here, based just on comparison with the shape, we cannot say whether we have bending moments here at this ending sides of this object, or we do have forces that are pointing towards the center. Anyway, this is how we explain the shape that is experimentally observed, and we could also make some predictions related to the shape here and the forces that are here at the pole. So if this is generated by pushing forces, this pushing force should be around 30 piconewtons, whereas here we predicted the forces around 300 piconewtons. So far about something what we observed in cells by doing microscopy, but this theory can be calculated without doing some experiments. So one can say, okay, we can also have a moment here that is in this direction, the same here, but in contrast to the moment in the first figure here, in the second figure, we have moment that is in the opposite direction. And if we have this opposite moment, then we'll have completely different shape. We'll have shape, we call this S shape, in contrast to this C shape of these microtubules shown above. And what I want here to emphasize is these shapes are different, but what is more different is what happens on one side of this object. We have here we have rotational here that is clockwise, whereas here we have counterclockwise direction of the rotational force. What is so this cannot become this one, and vice versa. So we can have, if we are discussing this as two-dimensional shape, we can have these two rather distinguished behaviors. What we do have in real cells? In real cells, microtubules are attaining actually variety of shapes, and I've just wanted to point some fibers here. One can see here at this outer microtubule case C shape, but also at this in this case of the inner microtubule, a nice S shape can be observed. And something similar to S shape, but it's unclear actually what type of shape is this microtubule. Sorry, I just wanted to show you, to check that these microtubules indeed have these shapes I pointed here. Of course, there is already a variety of these shapes, and our question was, maybe these shapes are not two-dimensional, and maybe this meaning. Sorry, just wanted to point out once again what I have shown before related to S shape and C shape. Here, for C shape, we need this clockwise rotational force, whereas here, in the case of S shape, we need counterclockwise rotational forces. And it's unclear how in a single spindle mutually could be present counterclockwise and clockwise rotational forces. And the question was, maybe these rotational forces are not so either or, maybe they're three-dimensional rotational forces, and also maybe this spindle has a three-dimensional shape. And now what we did here in this imaging, we did imaging, but now in the vertical spindle, meaning our imaging plane is going here along these cross sections. And in the case of just planar microtubule shapes, this movie should have points moving outward, inward. Whereas here one can clearly see this rotational movement of these points, which actually tells us that we have a complex 3D shape in this spindle. Of course, to understand the forces here in this spindle, we wanted to build a model, and for building model, we also needed one additional experiments. So we wanted to exclude the forces that are coming from the cortex. So we perturbed this cell cortex. So though there is no forces that are coming from this side, from the cortex side, this spindle still has this shape that is twisted, meaning we still have helicity in the or twist in these spindles. So theory is now rather simple. We consider these bundles as a single rods. So each bundle is a single rod. And here I already simplified everything to have only two rods. And in this case, if I have two rods and these two rods are extending from one pole to the other, I can write down force balance equations and balance of torques also for each of these objects. So we have four objects. We need balances for these two microtubules and also for these two spheres. The key part of the model is proper description of the bending and the twisting moment. So if we have some general bending and twisting moment acting on a fiber, then actually we need to describe it by more general equation. So Euler-Bernoulli approach is not appropriate anymore. And so we have now this static Kirchhoff equation. And this static Kirchhoff equation has these two parts. One is the bending part and the other is twisting part. And when we calculate the shapes by using this model, we could get actually variety of shapes. We could get this C shape. We could get some shape that is that extends in 3 dimensions. So this is, so just to explain what one can see here is in blue is shown y axis versus this main axis, whereas in white is shown the other z axis versus this main x axis. So as shown here in this drawing. And what one can see from these results is that we have as two extremes, we have C shape and S shape, but between these two shapes, we can also see these shapes that extend in 3 dimensions that are not pure C shape or pure S shape. And to show how one can see these shapes if it's just if it's watching from one side to the other, this is shown in this YZ projection. In this YZ projection, these 3-dimensional shapes are now getting these shapes that are having this petal-like shape. And indeed, these shapes are in agreement with the observed shapes. All shapes in the cell can be explained now with this general theory, where I didn't need to introduce something what I didn't like, what I have shown at the beginning of my talk, meaning this counterclockwise bending moment. So one can really explain this variety of shapes by having this bending moment and slight twisting moment present in the spindles. Of course, one can ask what type of molecules can exert twisting moment. It is shown that in vitro, Kinesin 1 can exert twisting moment of size that is 1.65 piconewtons times microns, and thus we will need 10 to 100 molecules, motor proteins, for generating such shapes, what is quite reasonable number. Of course, there is also a question whether we can understand the generators of this helical shape, and to get there, we were checking one in vitro study. So this is the study in which authors have surface coated by Kinesin 5 molecules, and they have these microtubules that glides on these motors, and they have this probe. And what the authors found that this probe is moving to the right here, and while it's moving, this probe was also rotating in a helical manner. So this Kinesin 5 is indeed one candidate for generating twists in spindles. Our collaborator, here I means Eva and her colleagues, inhibited Kinesin 5 by treating cells with these STLC molecules, and they found reduced helicity in these cells. So here just to conclude this part of my talk, so the overall shape of this spindle should be considered not only as this spindle shape, but also should have this. It also have this chirality. We could explain such shape by having torques, twisting and bending moments in the spindle, and we also found that this torque can be generated by kinesin proteins. Here, just before ending, I would like to thanks my colleagues working on these projects. So these are theoreticians working on these two projects. Here are experimentalists working on these two projects. I also would like to thanks to the rest of my group, and finally to your attention.
Thanks, Nenad, for the very nice talk. We can take more questions now. I think Vijay has a few questions for you. Go ahead, Vijay.
Yeah, thanks, Nenad. Very nice talk. So a couple of questions.
Thank you.
So first one, I mean, I wonder if the twist direction of the microtubule filament that you showed has some connection to the intrinsic chirality of the way the tubulin monomers themselves are arranged along the filament. I mean, so I asked the following question, that you showed that the filament curves one way, twists one way, so it gives you a net chirality. Is it always like that? I mean, you don't get opposite chirality sometimes?
This is an overall conclusion. However, it is not, so this chirality is not so conserved in sense there is a variability between cells. So if you check different cells, you will get different twist. And so there is indeed huge variability in this twist among cells.
Okay,
and,
so it's not the chirality of the filament itself that is setting this.
It's not the chirality of filament itself. So based on this study, I would say the chirality of single filaments leads to chirality of the entire spindle. So we indeed see chirality or this twist of a single filament, but also of the spindle as one object.
Okay, so the second question.
So we do see both.
I see. Okay. So the second question is, since you have this set of filaments which are sort of chiral, do you actually see a net rotation of the spindle about its axis?
That's a really interesting question. So far we didn't see rotation of the spindle. Sometimes, however, sometimes rotations can occur, but not of the spindle. Sometimes we saw rotations in the case where the spindles are destroyed and only monopoles are present. So if you have monopolar case, you will see this aster-like behavior, but you can also see something, some shape and rotation like a galaxy is rotating.
Okay, thank you.
Thanks. There's another question from Yash. Go ahead, Yash.
So you said like the astral microtubules don't contribute much, but can the chirality from the central spindle be extended or projected towards the astral filaments? Like, do they eventually twist because they are not bundled, they are mostly single filaments? So
sorry, I'm not sure that I understand completely your question. So there is, I will now try to answer what I think that you were asking. So there is a pulling force from the cortex, and this cortex also can do some rotations. And because of this rotation, actually pulling from cortex can generate rotational forces within the spindle. And I'm not sure if this is your question, but this is something what is already shown, that that there is rotation of the cell cortex. So there are nice papers from Stefan Grill lab, and you can check this chiral shape, chirality in this cell cortex behavior. And of course, if you have microtubules that are bound to something that that rotates, these microtubules could also generate rotations within the spindle. But we are not sure if this is the case in our spindles. Did I answer your question? I'm not sure that I completely understood your question.
I think you pointed me out. I can check that literature. But so basically, the main question was the center of the chromosome's single pole is like connected on the both sides. One side is the cortex, another side is the another pole. And then
Yes.
you start twisting one side, does it affect the other side? Like,
oh, is there a mechanical link between these microtubules and microtubules that are now other microtubules? Well, yeah. I think there is a link. We have some. I mean, there are some reasons why we can say that there is a link, because some perturbations, like what Eva have shown, these laser cutting experiments, sometimes these microtubule bundles are moving together, and also there is a paper from McCain's lab where they have shown this. The title is a bit misleading. It's called non-autonomous movement, but what they have shown, that this kinetochore movement and kinetochore oscillations in the neighboring fibers are correlated. So this can be only if there is a link between these neighboring fibers. Is this link at the pole or along the microtubule length? It's not completely clear to me, but there is a link between microtubule fibers. They are not completely independent.
Thanks, Nenad. I think there's another question from Vijay. Go ahead, please, Vijay.
Yeah, hi. Nenad, since you mentioned the cortex chirality, I mean, this is just a speculation. If you look at the establishment of the left-right axis in C. elegans, for instance, the chirality of the cortex is something that is supposed to play some part. The chirality of the spindle, I never thought would actually show up in that level. But do you know, for example, at least in the C. elegans zygote, when the LR axis is established at the 6-cell stage, the spindle is also chiral?
I think not. I think that these spindles in C. elegans they haven't shown chirality. So I was checking these papers on electron tomography papers, and these EM data have shown that chirality can be observed in these cells, like these human cells or some other cells, but I'm not aware that they see chirality in C. elegans cells.
But the mechanism seems quite general, and the model that you wrote down is rather generic physical description. So wouldn't you expect that to show up anywhere?
Yeah, it's general to a large extent, but this spindle in C. elegans is rather different compared to the other spindles. They have one huge kinetochore, and it's really very different. I think it's not so surprising that it's not completely that this mechanism is not completely present. What I didn't show here, but it's also interesting, spindles in yeasts are also chiral. So they also have this helical shape. Maybe I can find some. I hide some slides just to not overwhelm you with too many data. But so this is oh, okay. I cannot show it that way. So do you see my
yeah,
station?
I do.
Okay. So here this is shape of two yeast spindles, and it is well documented that they indeed have this twisted shape. This is not my interpretation. This is also interpretation from the authors. So these twisted shapes are observed within single rod spindles of fission yeast or budding yeast. Well,
Thanks.
So many nice questions. I'm so
Thanks
happy to
once
hear
again, Nenad, for the great talk. I think we've had two very excellent talks to kick off the meeting, and we're looking forward to the rest of the talks for the next few days. I think we can end today's session here. Thanks once again, Eva and Nenad. And as always, please do write in the Slack channel if you have any more questions for Eva or Nenad. Thank you, everybody.